Hi everyone, welcome to MR Learning Circle. So this is the 16th lecture of our microbiology MCQ series, and in this lecture we are going to cover the chemotherapy and the antibiotics section. So if you have not watched the previous videos, please go and watch it. It is very important for your upcoming GATE examination, and to find the link you can get the link above here or in the description box. And to get the copy of your ebook of your microbiology notes and the MCQ and the multiple select question, you can find the link in the description box. Okay, so now let's start with it. So the first topic is general characteristics of antimicrobial drugs. First question is, selective toxicity of an antimicrobial drug is primarily determined by, your options are A, ability to inhibit all microbial enzymes, B, targeting structures or pathways present in microbes but absent in host, C, broad spectrum activity, D, high drug concentration. So the correct answer for your question is option B, targeting structures or pathways present in microbes but absent in host. So what is selective toxicity? It relies on exploiting differences between microbial and the host cell. So here the example you can take is bacterial cell wall synthesis. Okay, now second question. A bacteriostatic drug. Options are A, kills bacteria immediately, B, inhibits bacterial growth relying on host immunity, C, works better in immunocompromised patient, D, targets viral replication. So the correct answer for this question is inhibits bacterial growth relying on host immunity, option B. So what does bacteriostatic drugs do? They stop the growth, but they do not kill. Okay, so host immune system clears the infection. It is stopping the growth, but it is not killing. Now question number 3. The spectrum of activity of an antimicrobial drug refers to. Options are A, its ability to penetrate tissues, B, range of microorganism it can inhibit or kill, C, time required to kill bacteria, D, mechanism of resistance. The correct answer is B, range of microorganism it can inhibit or kill. So narrow spectrum drugs target specific pathogen, whereas broad spectrum drugs, they affect multiple types of microorganism. Okay, now question number 4. Minimum inhibitory concentration, that is MIC, is defined as. Options are lowest concentration of drug that kills 99.9% of bacteria, B, lowest concentration that inhibit visible growth, C, concentration causing toxicity in host, D, standard dose for all infections. So the correct answer for this is option B, lowest concentration that inhibits visible growth. Question 5. Bactericidal drugs are preferred in options A, mild infection, B, immunocompromised patient, C, skin colonization, D, viral infection. Options correct answer for this is immunocompromised patient. So bactericidal drugs directly kill the bacteria, which is critical in patient with weak immunity. So all the bacteria would be killed, even the good ones. Okay, so that's why. Now question 6. Synergism between antimicrobial drugs is best exemplified by. Options are A, penicillin and tetracycline, B, trimethoprim and sulfonamide, C, erythromycins and chloramphenicol, D is aminoglycosides and chloramphenicol. So correct answer is B, trimethoprim and sulfonamides. So TMP and sulfonamides, what do? They block the sequential steps in folic acid synthesis and enhancing effect. So that's it. Question 7. The term post-antibiotic effect refers to. Options are A, bacterial growth inhibition persisting after drug removal, B, rapid bacterial regrowth after therapy, C, drug-induced host toxicity, D, resistance development. Correct answer for this is option A, bacterial growth inhibition persisting after drug removal. Now question number 8. Some important pharmacokinetic factor influencing antimicrobial therapy is. Options are A, MIC only, B, absorption, distribution, metabolism and excretion, C. Resistance only. D. Toxicity profile only. So the correct answer for this is option B, absorption, distribution, metabolism and excretion. So pharmacokinetics, it determines the drug levels at infection sites and dosing regimes. Okay. Now next is question 9. Broad spectrum antimicrobial can lead to. Options are A. Targeted therapy, B. Resistance development and disruption of normal flora, C. Enhanced selective toxicity, D. Increased host immunity. So the correct answer for this question is resistant development and disruption of microbial flora. So broad spectrum drugs, they may kill the commensals and promote resistant strains. Now question number 10. The sterility assurance level of antimicrobial treatment refers to. Options are time required to kill spores, B. Probability that an item remains non-sterile after treatment, C. Drug doses required, D. Drug stability. So B. Probability that an item remain non-sterile after treatment. So SAL quantifies the probability that a sterilization method leaves a viable microorganism. Okay. Now next is antibiotics, classification, mechanism and resistance. So the first question in this is, beta-lactam antibiotics primarily act by A. Inhibiting protein synthesis, B. Disrupting cell wall synthesis by binding penicillin binding proteins, C. Inhibiting DNA replication and D. By disrupting membrane. So correct answer is option B, disrupting cell wall synthesis by binding penicillin binding proteins. So what do beta-lactams do? They prevent the peptidoglycan cross-linking and that causes the lysis of the cell. Okay. Question number 2. Aminoglycosides exert their effect by. Options are A. Inhibiting 30S ribosomal subunit, B. Inhibiting folic acid synthesis, C. Disrupting membrane, D. Inhibiting DNA gyrase. So the correct answer is by inhibiting 30S ribosomal subunit. So aminoglycoside, they bind to 30S subunit and this causes the misreading of the mRNA and the production of the defective proteins. Now question 3. Tetracycline inhibit bacterial growth by. Options are A. Binding 50S ribosomes, B. Binding 30S ribosomes and blocking tRNA attachment, C. Inhibiting DNA replication, D. Oxidizing cell membrane lipid. Correct answer, easy question. Option B, by binding 30S ribosomes and blocking tRNA. Now question 4. Quinolones resistance commonly occur due to. Options are A. efflux pumps and mutation in DNA gyrase, B. beta-lactamase production, C. ribosomal methylation, D. cell wall thickening. Okay, so the correct answer for this is option A, efflux pumps and mutation in DNA gyrase. So mutation in DNA gyrase A and DNA gyrase B or efflux pump leads to the overexpression. Overexpression reduces what? Quinolone effectiveness. Okay. Now the next question, question 5. Macrolides inhibit bacteria by. Options are A. Disrupting membrane, B. Binding 50S ribosomal subunit and blocking translocation, C. Inhibiting DNA gyrase, B. Inhibiting folate synthesis. So correct answer is option B, and this is because they prevent the elongation of the protein chain. Okay. Question 6. Vancomycin resistance in enterococci is primarily due to. Options are A. beta-lactamase production, B. modification of D-alanine target to D-alanine D-lactose. So C is efflux pumps, D is sporin mutation. Okay, so correct answer is B. Question 7. Sulfonamides inhibit bacteria by. Options are A. Inhibiting dihydroapterate synthetase in folic acid synthesis, B. by binding 50S ribosome, C. by disrupting cell wall, D. DNA intercalation. So the correct answer is A. Inhibiting dihydroapterate synthetase in folic acid synthetase. So sulfonamides are competitive inhibitors of PABA in folate synthesis. Now question 8. Beta-lactamase inhibitors, example calvininic acid, are used with penicillin to. Options are A. enhance absorption, B. inhibit bacterial beta-lactamase and restore activity, C, reduce toxicity, D, broaden spectrum. So the correct answer is inhibit bacterial beta-lactamase and restore activity. Question 9. Linezolid acts by inhibiting 30S ribosome, binding with 50S ribosome and preventing formation of initiation complex, C, by inhibiting DNA gyrase, D, by disrupting membrane. So correct answer option B, binding 50S. So prevents the formation of functional 70S initiation complex, and it is effective against your MRSA. Now question 10. Mechanism of multi-drug resistance in bacteria include, options are enzymatic inactivation, B, target modification, C, flex pump, D, all of them. So the correct answer is all of the above. Now next topic is antifungal and antiviral drugs. Question 1. Amphotericin B kills fungi by, options are A, inhibiting ergosterol synthesis, B, binding ergosterol and forming spores in the membrane, C, inhibiting DNA synthesis, and B, by disrupting cell wall. Correct answer is option B, binding ergosterol and forming pores in membrane. Now question 2. Azoles inhibit fungi by, options are A, by binding ergosterol directly, B, by inhibiting 14 alpha D methylase in ergosterol synthesis, C, by disrupting cell wall, D, by inhibiting protein synthesis. Correct answer for this is inhibiting 14 alpha D methylase in ergosterol synthesis. Question 3. Flu cytosine acts by A, inhibiting RNA and DNA synthesis via conversion to 5-fluorouracil, B, disrupting ergosterol, C, binding tubulin, D, disrupting membrane permeability. So the correct answer is option A, inhibiting RNA and DNA synthesis via conversion to 5-fluorouracil. So flu cytosine is converted intracellularly to 5-fluorouracil, and that interferes with nucleic acid synthesis. Now question number 4. Acyclovir, an antiviral drug, is activated by, options are A, viral thymidine kinase, B, host DNA polymerase, C, reverse transcriptase, D, protease enzyme. So the correct answer is option A, viral thymidine kinase. So acyclovir is phosphorylated by viral thymidine kinase, and that inhibits viral DNA polymerase. Okay. Question 5. Oseltamivir, that is Tamiflu, inhibits, options are A, viral RNA polymerase, B, viral neuraminidase, C, reverse transcriptase, D, protease. So correct answer is option B. Question number 6. Resistance to acyclovir occurs primarily via, options are A, mutation in viral thymidine kinase, B, efflux pump, C, increased DNA repair, D, ribosomal modification. So correct answer is option A, mutation in viral thymidine kinase. Question number 7. Grisofulvin treats fungal infection by, options are inhibiting mitosis via microtubule disruption, B, disrupting membranes, C, inhibiting DNA synthesis, D, inhibiting ergosterol synthesis. So the correct answer is option A, inhibiting mitosis via microtubule disruption. The grisofulvin, it binds with tubulin, and which inhibits the fungal mitosis. Now question number 8. Ribavirin is active against, options are A, DNA virus, B, RNA virus, C, fungi, D, bacteria. So the correct answer is option B, RNA virus. Question number 9. Echinocandins inhibit fungi by, options are, so echinocandins Inhibit fungi by options are A. Blocking beta 1,3 D glucan synthesis in cell wall, B. Disrupting membrane, C. Inhibiting protein synthesis, D. Inhibiting DNA synthesis. So correct answer is option A. Blocking beta 1,3 glucan synthesis in cell wall. Now question number 10. Protease inhibitor in HIV therapy act by options are A. Inhibiting viral DNA polymerase, B. Preventing cleavage of viral polyproteins, C. Blocking reverse transcriptase, D. Inhibiting viral entry. Correct answer option B. Preventing cleavage of viral polyproteins. So protease inhibitor, they prevent the maturation of the viral proteins, producing a non-infectious virion. So that was all for this lecture. I hope you have learned something from this. And if yes, then please like the video, share it with your friends and do comment down below. Thank you for watching. Keep studying.